Congratulations, everybody. You have reached the last topic that we will cover in this physiology course, and it deals with the urinary system. What I've done is I've broken it up into, again, 2 mini lectures to kind of split it up into manageable chunks. This one is going to go over a little bit of the anatomy of the urinary system, just as a review, but we're going to concentrate on things like the blood supply to the kidneys. We're going to take a look at the overall structure of the nephron. And then we'll get into, in the next mini lecture, a little bit more complex topics where we take a look specifically as to what the nephron does in the formation of urine. Here's an anatomy review slide for you guys to take a look at. Again, since you're anatomy graduates, you should basically know what these anatomical structures are and basically what they do.
While we're not going to be covering this in physiology, I do want to remind you that the kidneys are located retroperitoneally. So they are found retroperitoneal, meaning behind the peritoneum. This means they're not part of the peritoneal cavity. And if you remember in our digestive lecture, I told you that the peritoneal cavity was kind of like a big Ziploc bag that contained all of the digestive organs. Well, the kidneys are found behind that Ziploc bag. The kidneys are found surrounded by 3 layers of tissue. The deepest layer is known as the renal capsule. This is a transparent sheet of dense, irregular connective tissue. Then you're going to add on to that a nice big thick adipose capsule. You can see right here the adipose tissue right there shown in the figure. It's called renal fat. This middle layer or adipose capsule is to protect the kidney from physical damage. And then there's an outer layer called the renal fascia. When you took anatomy, you learned that the kidney was divided internally into an outer cortex and an inner medulla. Now I've drawn a white line here in this model of a kidney to denote the outer cortex, which would be right here, and the inner medulla, which would be right there. The reason why we have to know outer cortex and inner medulla is that you have to know it in reference to nephrons. There are going to be nephrons that are called cortical nephrons, and there are those nephrons that are going to be called juxtamedullary. And it references the position of the glomerulus, the filtration structure of the nephron, where it is in that outer cortex. The medulla consists of 8 to 18 cone-shaped regions known as renal pyramids. I've circled a few in yellow. The wider base of the pyramid faces out towards the cortex, and then there is a narrow region known as a renal papilla, which projects down into the medulla. The regions of the medulla in between these pyramids are known as renal columns. The renal cortex can be divided into 2 zones, an outer peripheral cortical zone and a deeper juxtamedullary zone. So in this figure, I've drawn a little dotted yellow line. And if you look at the outside of that I've just diagrammed here in red, that is the cortical zone. And I'm going to put a little star here in green. That's the juxtamedullary zone. And as I mentioned in the previous slide, this relates to 2 specific kinds of nephrons, a cortical nephron and a juxtamedullary nephron. And they are classified this way based on the location of their glomerulus, which is the filtration part of the nephron. If the glomerulus is located in the cortical zone, this is a cortical nephron. And if the glomerulus is located in the juxtamedullary zone, this is a juxtamedullary nephron. Don't be afraid of the word juxtamedullary. Juxtamedullary is a fancy way of saying adjacent to the medulla.
Hopefully you remember that the renal papilla, which I'm going to diagram here, right here, this renal papilla, projects down into a cup-like structure called a minor calyx. So this structure right here, this cup structure, is called a minor calyx. The job of the minor calyx is to collect the urine that is coming out of the renal papilla. 3 or 4 minor calyces will unite to form a major calyx. I'll put a dot right here where the major calyx would be. And then all of those major calyces, maybe 2 or 3 of them, unite to form a renal pelvis, right there. The renal pelvis narrows out to form the ureter.
The job of the kidneys is to filter blood. So it has a very extensive and well-described blood supply. You learned many of these arteries and veins when you took anatomy, but let's remind ourselves. Each kidney is going to be supplied by a renal artery. This renal artery is going to divide into large arteries called segmental arteries. These segmental arteries are going to supply segments of the kidney. The segmental arteries are going to give off smaller branches that are going to pass through the renal columns. These are known as interlobar arteries. And then they are going to arch up over the top of a renal pyramid as arcuate arteries. Small blood vessels, small arteries, will branch off of the arcuate artery and run into the cortex of the kidney. These arteries are known as interlobular arteries. So the interlobar arteries run through the renal column. The interlobular arteries branch off of the arcuate. The interlobular arteries are then going to branch one more time, and they're going to give rise to small arteries known as arterioles. Specifically, they're going to give rise to what are called afferent arterioles. Here is an afferent arteriole right here, leading into a tangle of capillaries called a glomerulus. The glomerulus is a capillary network, and the glomerulus is the site of blood filtration. It is going to be surrounded by the very first part of the nephron, shown here in this picture in yellow. The very first part of this nephron is called the Bowman's capsule. So we have blood coming in the afferent arteriole, flowing into the glomerulus, and then the blood is going to come back out. And it is going to come back out to form the efferent arteriole. So if you get confused, just think E stands for exit. This blood that is coming out of the efferent arteriole, or what I should say is the efferent arteriole, is then going to form an extensive capillary network that is going to lay down on top of the rest of the nephron so that the nephron can reclaim what's been filtered out and dump what we want to keep back into that capillary network. The glomerulus is going to be drained by an efferent arteriole. The efferent arteriole is going to form an extensive capillary network that's going to cover and surround the tubules of the nephron. As I said in a previous slide, it's called the peritubular capillary network. There are some nephrons, however, in which this network covering the lower portion of the nephron is given a different name. It's given the name vasa recta. We're going to see specifically the vasa recta is the peritubular capillary network that is found on juxtamedullary nephrons. It's not really important anatomically if you know what the difference is between a peritubular capillary network and a vasa recta. Basically, they are all connected to one another and they're all the same thing, an extensive capillary network that lays on top and surrounds the tubules of the nephron. This is again because the kidney overfilters our blood plasma, and we have to reclaim close to 90% of what we have filtered out. And that's the job of the nephron in this capillary network. The cells lining the nephron are going to reclaim what we want to keep and going to dump it into this capillary network on top of the nephron. That capillary network will eventually form the interlobular veins, the arcuate veins, the interlobar veins, the segmental veins, and then that filtered blood is going to exit the kidney as the renal vein. So what is this nephron I've been talking about? A nephron is a series of tubes covered by that capillary network. The job of the nephron is to reclaim what we have filtered out of the blood and put it back into the bloodstream. So the nephron is a filtration and reclaiming structure. There are about 1 million nephrons found between the two kidneys. And here is an astounding amount. The kidneys filter 180 liters of blood per day. Now, we only urinate out a couple liters, so you can see just by those numbers that the job of the kidney is to reclaim most of what we have filtered. Each nephron essentially is a renal corpuscle, which is going to be the glomerulus and its Bowman's capsule, and then a series of interconnected tubules. Think of the renal corpuscle as being the filtration unit of the nephron. As I just said, it's the glomerulus surrounded by that glomerular capsule, which is also called the Bowman's capsule. And the rest of the nephron is known as the renal tubules. This is going to be for the reabsorption of what we have filtered out. It is going to be where we reabsorb water, ions, anything else that we have filtered out that we want to reclaim back into the blood.
So here's a very nice, very simple picture from the anatomy slides that I've shown of the nephron, giving you the parts of the nephron: the Bowman's capsule, the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and the collecting duct, which is going to collect from multiple nephrons. I am going to short form the proximal convoluted tubule as PCT. I'm going to short form the distal convoluted tubule as DCT. Will you short form these when you are writing your answers or doing your exams? No. You have to write out these structures. I get to write them as short forms. The power of being the professor, I guess.
I've used this slide in anatomy for years to just very simply describe the path of filtrate through the nephron. First of all, what is this term filtrate? This filtrate is exactly as its name implies. It is what we filter out of our blood plasma. So think of filtrate as pre-urine. Blood is going to enter into the glomerulus. It is surrounded by the Bowman's capsule. We're going to do an extensive amount of filtration, and that fluid is going to move into the Bowman's capsule, and it is known as filtrate. That filtrate will then flow through the proximal convoluted tubule, down into the medulla through the descending arm of the loop of Henle. It'll do a little U-turn. It'll come back up through the ascending limb of the loop of Henle. It'll then flow through a distal convoluted tubule, and then the distal convoluted tubule will turn around and connect up to form the collecting duct. Multiple nephrons are going to connect up to a single collecting duct. And pretty much what you have at the bottom part of the collecting duct is urine. That urine is going to leave the collecting duct. It's going to enter into a little cup-shaped structure called a minor calyx. A bunch of minor calyces are going to fuse to form a major calyx. The major calyces in our kidney are going to form the renal pelvis. The renal pelvis narrows and exits the kidney as the ureter, and the ureter then takes that urine down to the bladder for storage. Before we leave the slide, I do want to point out one thing to you. Please note that this peritubular capillary network lays on top of the nephron. Again, a lot of anatomy textbooks will call this portion, which sits on the superior portion of the nephron, the peritubular capillary network, and it often calls this portion, which sits on the loop of Henle, the vasa recta. I'm really not that picky. If you want to call the entire thing the peritubular capillary network, I'm fine. But this is where reabsorption occurs in the nephron. The cells lining this nephron are going to reclaim what we want to keep and move it out of the filtrate and across into the blood in this peritubular capillary network. This capillary network will then take all that reclaimed material and it will flow eventually out of the kidney via the renal vein.
About 80 to 85% of our nephrons are called cortical nephrons. The reason why they are called cortical nephrons is pretty simple. The actual renal corpuscle, the glomerulus and Bowman's capsule, is found in the cortical region of the cortex. So I'm going to put a dotted green line there. Above that is going to be the cortical region of the cortex, the most superficial region of the cortex. You can see that these nephrons have a very short loop of Henle. The loop of Henle is mainly in the cortex, but there is a portion that kind of dives into the renal medulla. This, I'll draw the dotted back line here. That's the dividing line between cortex and medulla. So there is a little bit of the loop of Henle that pokes down into the medulla. And the last thing you'll probably notice is there's a pretty extensive capillary network. Cortical nephrons are the nephrons that form our ordinary average everyday urine. They're the majority of the nephrons and they form the majority of the urine that we make.
The remaining 15 to 20% of our nephrons are known as juxtamedullary nephrons. So let me put some dotted lines back in here. I'm going to put the dotted green line here, which represents the border between the cortical region of the nephron and the juxtamedullary region of the nephron. And then I'm going to use my black line here and I'm going to put in the border between the cortex and the medulla. The reason why I'm doing this is because I want you to see that the renal corpuscles of a juxtamedullary nephron are located in the deeper region of the cortex. They're located in that juxtamedullary region. Do not be afraid of the term juxtamedullary. To juxtapose something means to put something on top of something else. So this renal corpuscle is on top of the medulla. That's where the name juxtamedullary came from. These nephrons have very long loops of Henle that extend very deeply into the medulla. And the reason why is these nephrons are responsible for tweaking the composition of our urine to produce either dilute urine or concentrated urine. You can see from this picture that the loop of Henle is covered with a capillary network that looks very different to the peritubular one. It's made up of these sort of little strings that almost forms a ladder-like structure. Now, this portion of the capillary network in these nephrons is called the vasa recta. It is still connected to that peritubular capillary network. It's just in anatomy, because it looks so different, it's given a different name. You may have learned in anatomy that the vasa recta is the part of the capillary network that covers the loop of Henle, and that's fine. But you can see where its name came from because it looks very distinct in the juxtamedullary nephron. I love this picture that I found on the web for you because it does show you two different nephrons. It shows you the cortical nephrons with their renal corpuscles in the more peripheral part of the cortex, and it shows you the juxtamedullary with the renal corpuscle located in the deeper part of the cortex. And it also shows you the peritubular capillary network covering the nephrons here, the cortical nephrons, and it shows you the difference between the capillary network found covering the juxtamedullary nephron. So you can see here in the cortical nephron, we have an extensive peritubular capillary network covering the entire nephron. And in the juxtamedullary nephron, you can see how this capillary network looks a little different. And specifically right here, the portion covering the loop of Henle looks very different. So anatomically, it's given the name vasa recta.
The average intake of water of a typical human being in a day is about 2.5 liters. 60% of that comes from drinking water, 30% from moist foods, and we actually are metabolically capable of making water. Only about 10% of the water we have in a day comes from that. So obviously, in order for us to remain healthy and in homeostatic balance, we have to drink. The regulation of intake of fluids is done by a group of neurons, a nucleus located within the hypothalamus. It is the thirst center. Don't ask me what hypothalamic nucleus it is, I can't remember. But what it does is it senses the osmotic pressure of blood flowing through the hypothalamus. And as our body loses water, as we become dehydrated, the osmotic pressure of our blood plasma goes up. And the osmoreceptors in this thirst center nucleus will detect the sudden increase in osmotic pressure within our blood plasma. And it will signal to us to drink. It will also signal to the posterior pituitary and tell the posterior pituitary to release the hormone antidiuretic hormone. Antidiuretic hormone is made by the hypothalamus. It's stored in the posterior pituitary. And what antidiuretic hormone does is it acts on the kidney, it acts on the nephrons of the kidney, and it acts on the distal convoluted tubule and the collecting duct of the kidney. And it tells those two areas of the nephron to increase their reabsorption of water so that we decrease our urine output. So that essentially our urine is a lower volume and it's more concentrated because we're not urinating out that normal amount of water. The more water that we can reabsorb from our body, from our kidneys, and put back into the blood, the more water we can reabsorb by drinking and absorbing it across our GI tract. This means putting that water back into the blood and decreasing our blood's osmotic pressure back into the normal range. Once we hit normal range, it's a negative feedback mechanism and it shuts off the release of antidiuretic hormone. If you drink too much water and you get too much of a drop in your osmotic pressure, this is going to tell your kidneys to increase their filtering activity and make a high volume dilute urine. And that's when the juxtamedullary nephrons come into play. A significant drop in the osmotic pressure of our blood can stimulate the juxtamedullary nephrons to increase their activity. So we end up urinating more than we normally do. So instead of 2.5 liters of urine a day, maybe we make 3 or 3.5 liters of urine a day. Hopefully you realize in order to remain in water balance, if you take in 2.5 liters of water, then 2.5 liters of water must be lost. 60% of our water is lost in urine, 6% is lost in feces, 6% in sweat. But actually, close to a third of the water we lose is evaporating from the surface of our skin and across our lungs. When you breathe out, that air is warm, but it's also moist. However, I will emphasize that the primary means for controlling water loss is through urine production. The last thing I will say is that no, you do not need to take in 8 liters of water a day to remain healthy. That is a myth which has been perpetuated by companies like Dasani and Aquafina so that you will go out and buy 8 bottles of the water a day. To remain healthy, the average human being needs 2.5 liters of water a day.
Our excretory system, or urinary system, has 4 functions. The first function is maintenance of water-salt balance of our blood plasma. The second function is the maintenance of acid-base balance of our blood plasma. The third function is the excretion of nitrogenous wastes. And the fourth function is the secretion of hormones. Let's start off with maintenance of water-salt balance in our blood plasma. Water is going to be reabsorbed back into the blood plasma from the following areas of the nephron: the proximal convoluted tubule, the descending loop of Henle, the distal convoluted tubule, and the collecting duct. Most of that water reabsorption is going to take place in the proximal convoluted tubule and descending arm of the loop of Henle. However, under the influence of a specific hormone called antidiuretic hormone, or ADH, we can get a little bit more water reabsorbed from the cells that line the DCT, the distal convoluted tubule, and the collecting duct. Salt is going to be reabsorbed from the proximal convoluted tubule, the ascending portion of the loop of Henle, and under the influence of a hormone called aldosterone from the distal convoluted tubule and collecting duct. So if you're getting the sense that the activity of the distal convoluted tubule and collecting duct requires hormones, you're exactly right. The next mini lecture is going to go over a lot of this, but I'm just going to say for now that water reabsorption and salt reabsorption can have dramatic effects on the physical pressure of blood, in other words, our blood pressure, and also the osmotic pressure of our blood. Blood pressure and osmotic pressure are going to control the movement of fluid between the extracellular fluids like blood plasma, the interstitial fluid we find in our tissues, and the cytosol, which is the fluid of the cell. For example, increased blood pressure can physically drive the movement of fluid out of the blood plasma into a tissue. So it can drive movement of fluid out of the blood plasma in a capillary network into the interstitial fluid of a tissue. And once it's in the interstitial fluid, some of that fluid's going to move into the cells to become cytosol. However, osmotic pressure will cause fluids to leave things like the interstitial fluid and enter back into the blood plasma. So there is a very complex interplay between blood pressure and osmotic pressure and how they control the movement of fluid in our body.
Another one of the balances that the urinary function controls is the acid-base balance of our blood. Dissolving CO2 in the water of our cells or the water of our blood plasma is going to produce hydrogen ions and bicarbonate ions. If you take CO2 and dissolve it in the blood plasma, you will produce carbonic acid, and that will dissociate into hydrogen ions and bicarbonate ions. These hydrogen ions help control the acidity of our blood. We want to have just the right amount of hydrogen ions so that the pH of our blood plasma is around 7.5 to 7.6. Okay, I think that means you would need 10 to the minus 7.5 moles of hydrogen ions in every liter of blood plasma. The bicarbonate that we produce through this dissociation reaction is reabsorbed from our urine back into the blood. So we want to basically keep these bicarbonate ions because it's a major buffering component of our blood. If you shift the balance towards bicarbonate ion, if you have more bicarbonate ions, then that means they combine more hydrogen ions. They can soak up excess hydrogen ions by reforming carbonic acid. So one of the things our urinary system does is it tries to keep the bicarbonate ions and it gets rid of excess hydrogen ions. Remember, these components are going to be filtered out at the glomerulus. So our filtrate flowing through our nephrons is going to be filled with bicarbonate ions and hydrogen ions. Our nephrons will reclaim the bicarbonate ions because I think of them as being a proton sponge. They soak up excess hydrogen ions, which can be made by our body. And then our urine basically gets rid of the hydrogen ions that we do not want. A lot of the hydrogen ions that are expelled out in the urine combine with trace amounts of ammonia in our urine to form ammonium. This explains why our urine has the smell of ammonia. In addition to balancing things, our urinary system excretes things. Specifically, it's responsible for excreting metabolic wastes. And these metabolic wastes are nitrogenous. The three nitrogenous wastes that are filtered out at the glomerulus and expelled out by the urine are urea, creatinine, and uric acid. Urea is a byproduct of breaking down old amino acids. It's done by the liver. This amine group is removed when that amino acid is broken down. It unfortunately then promotes the formation of ammonia, which is very toxic. So the liver combines the ammonia with a carbon dioxide molecule to produce urea. So the blood now contains urea. It will contain a very small amount of ammonia, not enough to make us sick, but it is there. But the majority of this is now urea. It's going to be filtered out at the glomerulus and become part of the filtrate. We actually do reclaim a little bit of urea. The bottom section of the collecting duct is designed to reclaim a little bit of this urea. The majority of the urea is urinated out, but we do reclaim a little bit, and I'll tell you why. Urea is really good at maintaining osmotic pressure. So we keep a little bit of in our blood plasma so that we can maintain a normal osmotic pressure range. Creatinine is a nitrogenous waste, which is produced by the breakdown of creatine phosphate in muscles. Remember, creatine phosphate is a source of high energy phosphate bonds. So when we go back and we take creatine phosphate and we remove the phosphate and stick it back onto the ADP to make ATP, creatinine is produced. Creatinine is filtered out at the glomerulus and it becomes part of the urine. Creatinine content in urine is used as a diagnostic tool to let medical professionals know just how efficient your muscle metabolism, how efficiently your muscle cells are producing energy. Uric acid is the last nitrogenous waste, and it is formed when you break DNA and RNA nucleotides down. The thing about uric acid is that it's insoluble. And if enough uric acid will be made, it precipitates out in the blood plasma. And it actually can get concentrated in areas where blood flows very slowly. Like, for example, joints. The blood that flows through our joints is at a pretty slow velocity. So the precipitating uric acid can crystallize out there, and it inflames the joint, and it causes what's known as metabolic arthritis or gout.
Believe it or not, our kidneys also have an endocrine function in that they are capable of secreting hormones. The hormones that the kidney secretes are erythropoietin. They also secrete a hormone called renin. And if you think that vitamin D has hormonal action on bone production, then the kidneys actually play a role in the activation of vitamin D and the production of vitamin D as a bone building hormone. Let's concentrate on the production of erythropoietin by the kidney. The job of erythropoietin, otherwise known as EPO, is to stimulate red blood cell production by the hematopoietic stem cells we find in our red bone marrow. And you might be asking yourself the question, how on earth does erythropoietin production even remotely relate to red blood cell production? Let me start off by saying one more time that our kidneys filter 180 liters of blood a day. Your kidneys are the prime monitoring station for the physical and chemical composition of your blood. So, in other words, the first organ that's going to know that the oxygen carrying content of blood has been compromised is the kidney. The kidneys have the ability to detect the oxygen being carried by our blood. Therefore, they have the ability to detect the reduced oxygen carrying capacity of our blood. And the reason why our oxygen carrying capacity has gone down is because the number of red blood cells carrying that oxygen has gone down. So when less oxygen containing blood is delivered to the kidneys, they realize this. They secrete the hormone EPO into the bloodstream, And as that blood flows through our large bones like our femur and our humerus and flows through the red bone marrow contained within those bones, the hematopoietic stem cells respond to that EPO by producing more red blood cells. Those extra red blood cells get put back into circulation, and suddenly our oxygen carrying content of our blood goes back to normal.
If the blood flowing through our kidneys does not have enough physical pressure, in other words, if our blood pressure drops, the kidneys will know this. They will release a hormone called renin, and through what's called the renin angiotensin aldosterone system, it will lead to the release of aldosterone by the adrenal glands on top. This is a mineral corticoid made by the zona glomerulosa layer of the adrenal glands. So you see how I can turn around and take that and relate it back to a lecture we did how long ago? As a mineral corticoid, aldosterone increases reabsorption of salt by the kidneys. It will eventually lead to an increase in blood volume and blood pressure. How? We're going to see that in the last mini lecture.
The last mini lecture in this series is lecture 19B. It deals with the physiology of the nephron, and it is your last lecture in this entire course.